Hello everybody. Today in this video, we'll study about gametogenesis. So, under gametogenesis, we'll study about spermatogenesis and oogenesis. This is the first video in which we'll study about gametogenesis and spermatogenesis. We'll study about oogenesis in the part 2 of this video. Now, what is gametogenesis? Gametogenesis, it is the process of formation of gametes from the primordial germ cells involving the cell division, mitosis and meiosis is known as gametogenesis. So, gametogenesis is actually the formation of gametes, that is spermatozoa in males and ova in case of female, from the primordial germ cell involving the cell division, mitosis and meiosis. So, the gametogenesis involves two processes: spermatogenesis in case of males and oogenesis in case of females. So, spermatogenesis occurs in case of male as a part of gametogenesis, and oogenesis occurs in case of female body as a part of gametogenesis. So, now we have to understand what is primordial germ cells. So, what are primordial germ cell? Primordial germ cell are derived from the epiblast. So, epiblast, what is epiblast? During the development, we all know that there are two type of cells. The outer cell is known as trophoblast and the inner cell mass is known as embryoblast, and these embryoblast differentiate into epiblast and hypoblast. So primordial germ cells are derived from epiblast. So during the development, the migration of mesenchymal cell from the epiblast occurs. So these epiblast migrate to the endoderm of the yolk sac in the fourth week of development, and this epiblast finally reach to the indeterminate gonad by fifth week to differentiate into gametes. So this is all about the primordial germ cell from which the gametes are derived. Now, without any delay, now we'll study about spermatogenesis. So, what is spermatogenesis? Process by which spermatozoa are produced from spermatogonial stem cell involving the cell division, mitosis and meiosis is known as spermatogenesis. So similar definition like that of gametogenesis, but in this case, spermatozoa are produced from the spermatogonial stem cell. So what are the different steps in spermatogenesis? At first, the primordial germ cell develop into spermatogonia. So spermatogonia is diploid structure, hence it is represented by 2n. So the primordial germ cell, as we have discussed earlier, from where the primordial germ cell comes from. So primordial germ cell develop into spermatogonia. In the first step, this formed spermatogonia, which is a diploid structure, then yield primary spermatocyte by mitosis. So all the three structure, that is primordial germ cell, spermatogonia as well as primary spermatocyte, are diploid structure. So on further development, primary spermatocyte then yields two secondary spermatocyte by undergoing meiosis first cell division. That is, the formed primary spermatocyte undergo meiosis first cell division to differentiate into two secondary spermatocyte. We all know that meiosis cell division is a reductional type of cell division. So, from the diploid structure from the primary spermatocyte, which is diploid, the secondary spermatocyte, which is a haploid structure, is produced. So, this secondary spermatocyte, each secondary spermatocyte divide meiotically by meiosis second to form two spermatid. Meiosis first is a reductional cell division, whereas Meiosis second is same like the mitosis cell division. So each secondary spermatocyte undergo meiosis second or similar type of mitosis like cell division, giving rise to two spermatids. Later, spermatid develops into spermatozoa by spermiogenesis. So, this is all about the spermatogenesis, in which at first primordial germ cell undergo development to form spermatogonia, which undergo mitosis to form primary spermatocyte. Primary spermatocyte undergo meiosis to form two secondary spermatocytes, and each secondary spermatocyte undergo meiosis second cell division from spermatid. Now we'll talk about the spermiogenesis. We can better understand in this figure the primordial germ cell are dormant or inactive until puberty in case of males. So the primordial germ cell first develop into type A spermatogonia. Actually, the type A spermatogonia do not undergo active mitosis type of cell division. So it further develop into type B spermatogonia. In the type B spermatogonia, this up to here, this process is known as spermatocytosis. So type B spermatogonia then undergo DNA replication to form primary spermatocyte, that is from the type B spermatogonia. Primary spermatocyte is formed. So the primary spermatocyte then undergo synapses crossing over and chiasma formation to form the secondary spermatocyte. The primary spermatocyte is diploid whereas secondary spermatocyte is haploid, because up to here the mitosis cell division is involved in the formation of primary spermatocyte, whereas meiosis occurs during the formation of secondary spermatocyte. Now the secondary spermatocyte then undergo a series of developmental changes to form the spermatid. Both the secondary spermatocyte and the spermatid are haploid structure. So this is all about the spermatogenesis, in which single primary spermatocyte produces four spermatozoa up to the last stage of spermatogenesis. Now we'll study about what is spermiogenesis. Spermiogenesis, process by which spermatid become spermatozoan. So the spermatid undergo a series of developmental changes to form the spermatozoa. The process is known as spermiogenesis. So we have to understand what are the different changes that occurs during the development of spermatid into spermatozoa. During this process, the nucleus forms head, the Golgi apparatus from the acrosomal cap, the central divide to form axial filament of flagellum, and the mitochondria surrounds the axial filament and forms the middle piece. So we can see in this figure, this is the nucleus, this is the Golgi complex, this is the mitochondria, and the central we can see here. So the nucleus forms the head. In the successive stage of development, we can see the nucleus is slightly developing, and finally it forms the head of the spermatozoa, and the Golgi complex from the acrosomal cap. You can see here this Golgi complex undergoes a series of developmental process and finally forms the acrosomal cap. Now the central forms the axial filament. We can see here at first the central is small, then it slowly elongates to finally form the axial filament, and the remaining mitochondria form the middle piece of the sperm or the spermatozoa. This is the figure showing the process of spermiogenesis. So, as I have said earlier, type A spermatogonia do not undergo active mitosis. So, in order to undergo active mitosis, type A spermatogonia has to be developed into type B spermatogonia, which undergoes active mitosis to form primary spermatocytes. So, most important question, duration of the spermatogenesis is, it is 74 days. So, thank you for watching. For more informative videos, please subscribe Medicos 1.0. Thank you everybody.